This project focuses on understanding the effects of low oxygen and low pH on oyster growth in Chesapeake Bay, especially looking at how exposure to both of those stresses early in life can lead to effects later in life. In Chesapeake Bay, low oxygen and low pH go through cycles every day where they tend to be higher during the day and low at night or in the early morning. Our laboratory system makes it possible to study both cycles of low pH and low oxygen at the same time and separate them out, so we can have some oysters that are exposed just to low pH, some just to low oxygen, and some to both. Then we can study how those stressors work individually and also how they interact to shape things like oyster growth rates.
We purchased our juvenile oysters from Horn Point Laboratories. We put them in the lab and exposed them to 6 different treatments varying in severity, and the oysters experience these cycles 5 times a week for 8 weeks. Before we deployed oysters in the field, we glued them to tiles to individually track oysters, and we had over 1,500 oysters sent out into the field. It's important to look at these 2 stressors together because you get a more accurate picture of how real oysters in the bay are affected by both pH and dissolved oxygen cycling, instead of just focusing on one treatment and one constant exposure to that treatment.
Once we finished the in-lab portion of the experiment, we outplanted our oysters to the different sites throughout the Chesapeake Bay, and they stayed out in the field for 12 weeks. Once the 12-week period was finished, we collected the oysters and brought them back to our laboratory to analyze the growth measurements, which included getting shell length, calculating shell area, and getting dry weights for shell and tissue separately. We took another photograph of the individual tiles in the lab so we could track individuals' growth rates over time.
We are in the process of analyzing data. We expect to have results during 2018, and we'll share those results with both the aquaculture community and restoration community. We think the results of this project will help inform how oysters are raised early in life history to better condition them to survive when they're planted out into the bay.